So the title is Sarcoplasmic Reticulum and Mitochondria. Relationship is altered in early stages of cancer-induced cachexia. So just to put some data, the data in context, cancer cachexia is defined as a multifactorial syndrome characterized by an ongoing loss of skeletal muscle mass with or without loss of fat mass. And the syndrome cannot be fully reversed by conventional nutritional support and leads to progressive functional impairment. This definition was established on a consensus in 2011. And the pathophysiology is characterized by a negative protein and energy balance driven by a variable combination of reduced food intake and abnormal metabolism. The same consensus established that cachexia cancer cachexia specifically, can be divided stratified in 3 stages. Precachexia, that is characterized by a weight loss under 5%, anorexia, and some metabolic changes. And the second stage is cachexia, characterized by weight loss above 5% or body mass index under 20, sarcopenia, and weight loss above 2%. And this phase is specifically characterized by systemic inflammation. The third phase is called refractory cachexia, where the patient is unresponsive to anti-cancer therapy and unfortunately have a low life expectancy. In general, cachexia leads to decreased quality of life and life expectancy, and is present in around 40% of all cancer patients. Usually, preclinical studies are carried out with very different experimental cancer models. Some are more frequently than others, but most frequently the experimental models are stratified in advanced cachexia stages. So to summarize here what's important to us, cachexia, the predominance weight loss in cachexia is muscle loss. Sometimes leads to fat loss, sometimes do not. So in the last years, our group is interested in investigate oxidative modifications, oxidative stress modifications in muscle loss in cancer cachexia, and using a Walker 256 solid tumor. This paper published in 2010 showed us that 10 days after a solid tumor implantation, Walker 256 solid tumor implantation, MDA, a marker of oxidative modification to lipids, lipid oxidation, is increased in muscles of Walker 256 solid tumor. Here is specifically gastrocnemius muscle. And that treatment with alpha tocopherol, lipid-soluble antioxidant, can revert this parameter. The same was demonstrated in oxidative modifications to proteins. And also alpha tocopherol treatment could revert this damage to proteins. Interestingly, atrogene 1 expression, that is a marker of proteolysis in muscle, specific to skeletal muscle, is increased 5 days after tumor inoculation. So apparently is not coincident with the modifications to lipids or proteins. And also atrogene expression could be reverted by alpha tocopherol treatment. So we decided to better investigate oxidation to lipids using a more sensitive technique that is based on chemiluminescence detection. And this more sensitive technique in gastrocnemius could show us that more sensitive modifications to lipids are present 5 days after tumor inoculation, also in gastrocnemius muscle, and could be reverted by the treatment with alpha tocopherol. So this data showed us that oxidative modifications to lipids could be critical and coincident with markers of proteolysis. And then we decided to better investigate membranes modifications in muscles. In the last years, literature says that mitochondria is involved in cachexia progression, but most parts of membranes present in muscles are sarcoplasmic reticulum membranes. Around 70% of all membranes in muscle are sarcoplasmic reticulum membranes. So we decided to investigate, as mitochondria is connected to sarcoplasmic reticulum, we decided to investigate what happens with SR in cachexia progression. So we used firstly, an experimental design that involved a control group, a tumor group, the same Walker 256 solid tumor that we used in the previous work from 2010. And also we tested that treatment with a NAC, water-soluble antioxidant, to test if the results could be similar to the ones that we previously observed. And also we collected data in a very precocious, in a very early stage of cachexia, just 5 days after tumor inoculation, evaluating also soleus and ideal, different fiber muscles with the different predominance of fiber types. We performed a series of experiments that then involved presence to levels of proteins of SR proteins, EM for mitochondria, electron microscopy for mitochondria and sarcoplasmic reticulum, proteolysis, and also oxidative stress parameters. What I first saw is that 5 days after tumor implantation, the pre-cachexia stage was established around 2.5% of general weight loss. And gastrocnemius, EDL, and soleus muscles were responsive to cachexia. The predominance of muscle loss was, as literature usually point, predominantly glycolytic fibers. And this loss of glycolytic fibers could be reverted by treatment with an acetylcysteine. So investigating a little bit better, we repeated that expression of atrogen that we detected in the paper of 2010 in gastrocnemius. And the results confirmed the increased expression of atrogen 1 in gastrocnemius muscle of tumor-bearing rats. And in the same way that what happened with alpha tocopherol, and acetylcysteine was able to revert this increasing of atrogen expression. Not surprisingly, in ideal muscles, the levels of carbonylated proteins or oxidative damage to proteins, weren't detected, not even in ideal or soleus muscle, showing that there is something that happens before protein oxidation that could be induced proteolysis and atrogen expression. So with the help of Simona Boncompagni in the University of Chieti, we could verify what was happening in ideal muscle, in muscles of tumor-bearing rats, and with the treatment of NAC in what is related to mitochondria. As you can see in this panel in the column at left, the mitochondria tend to fission in tumor-bearing rats in ideal muscle, and treatment of neck with N-acetylcysteine tend to revert and reestablish some level of fission. And the quantitative analysis confirmed these results. The same tend to happen also in soleus in the same time, in the same periods of time, but the quantification is in progress at this moment. So when, with the help of Simona Boncompagni, we decided to investigate a sarcoplasmic reticulum by electron microscopy. In ideal muscle, we could observe that just after, only 5 days after tumor inoculation in the experimental model of a Walker 256 solid tumor in rats, the double layer of a sarcoplasmic reticulum practically disappear, as you can see in this arrow here. Some level of sarcoplasmic reticulum is reestablished after, with the treatment, not reestablished, but prevented with the treatment with NAC. When we quantified SR, we also observed that soleus muscle also lost SR membranes. So we decided to investigate how was the oxidative modification to membranes with the previously mentioned very sensitive method based on chemiluminescence to quantify lipid peroxidation or lipid oxidation. So in ideal muscles, we could observe that the data reproduced and are coincident with what we saw in electron microscopy. But in soleus, no, we do not observe. We have increased levels of lipid peroxidations in ideal, but it's not proportional. The data are not the same found in soleus. So in ideal muscles, we decided to investigate the proteins involved in excitation AC coupling or calcium reuptake. And in ideal muscle, we quantified calsequestrin 1 and CERCA1. Calsequestrin 1 demonstrated to be decreased in tumor-bearing rats and partially recovered by N-acetylcysteine treatment. And the other phenomenon could be observed in CERCA1 quantification with increased amount of CERCA1. And this amount of proteins were partially reversed by N-acetyl treatment, with the N-acetyl treatment. As an alternative to Western blotting, we decided to try an immunohistochemistry assay to verify RYR1 amounts also in ideal muscles. And we also verified that this protein that is critical to calcium handling is decreased in tumor bearing rats, and in the same way this decreasing is partially protected or prevented by NAC treatment. With the help of Laura Pietrangelo, we also could observe that calcium release units are decreased in tumor bearing rats. So there is some type of uncoupling in miscommunication between sarcoplasmic reticulum and mitochondria in these experimental models. So in base with these results, we decided to investigate if progress in the experimental models. As some tumor types have different metabolic behaviors, we try to compare, we are trying to compare what happens in precacheexia of other experimental, tumor experimental models. We are comparing at this point with Walker.
Sorry, 5 minutes left. Thank you.
Okay, I'm finishing. With we are comparing with 256 tumor type, the results with another breast tumor in mice, Ehrlich tumor. C26 mice is a colorectal cancer in mice, and B in a melanoma experimental model too with a B16F10 cells. We could, by means of pilot studies, we could see that in precacheexia, 14 days in Ehrlich tumor, 7 days in C26 tumor, and 22 days after in B16F10 tumor, we have precacheexia stages. In all of the stages, we have decreased ideal muscle, as literature usually says, but only in C26 and melanoma, we have increased protein oxidation. So glycolytic muscles are already prone to mass waste in precacheexia, but not necessarily dependent of protein oxidation. The second conclusion that we could reach until now is that these parameters are more correlated to oxidative modifications to membranes, as could be seen in ideal muscle of C26 and also Ehrlich breast cancer. In melanoma, the data is in progress, but we believe that the results are practically the same so far. The third conclusion in the second experimental design is that the oxidative muscles are not preserved from both damages. Solus have also decreased muscle mass, not in the same proportion of video, but they are also compromised. And lipid oxidation, oxidative modifications to lipids are present in these models. So the conclusions so far are that this data demonstrated that in early stage of breast cancer carcinoma-induced CACHEXIA, where body mass waste is not well established, EDL is already sensitive to mitochondria NSR dynamic alterations, which corresponds to oxidative damage to membranes, that probably participates of the skeletal muscle mass modulation before clinical cachexia comes to light. And in addition, it seems that these alterations appear in different periods of each tumor type, and oxidative stress is involved also in the process as sarcoplasmic reticulum and EC coupling. So I would like to thank all my collaborators, especially Claudia Pecorai, Laura Pietrangelo, Alessia Di Fonso, Simona Boncompagni, and Feliciano Protasi, obviously, from University of Chieti, my team, and I leave you with this beautiful picture from our university. Thank you, everybody, for the precious attention. Thank you.
Thank you, Flavia. Maybe you remember that we met some years ago in Padova.
Yeah, obviously.
We have 1 minute for one question.
1 minute for one question.
I'd just like to make a quick comment. I work with the ERG channel, and what we find is that it is upregulated in muscle of animals suffering with cachexia. And when we put it into the muscles, we also find that we can induce atrophy through a couple of different mechanisms. One of the mechanisms we have not at this point explored, although we have some next generation sequencing data that suggests
short question, please.
The ERG also upregulates reactive oxygen species producing enzymes. So it's something to think about in terms of mechanism of increasing reactive oxygenation species producing enzymes.
You mean salt catalase and these types of
I'm
enzymes?
sorry?
You mean salt, catalase, or this other type of enzymes? I'm not sure if I understood well your question.
Well, enzymes that, yeah, reactive oxygenation producing enzymes, superoxide, mutase. We've got a number of them. I could give you a list of it. I could share that data with you if you were interested.
Yeah. We have measured some enzymes, but at this moment we have no significant a great amount of data to produce a graph. But obviously this needs to be checked.